Hello everyone, my name is Miguel Cuesta and I work as a junior investigator at INIA CSIC in Spain. It's a real pleasure for me to share with all of you this interesting piece of work here in the third edition of World Congress on Virology and Infectious Disease. I'm going to share with all of you this pioneer information regarding the insights into the function of the ESCRT complex and LBPA in African swine fever virus infection. This is a virus, this is a lethal virus without no cure, and it affects swines, and therefore all the porcine industry is threatened by this virus. And the worst thing is that it's spread worldwide, so it's a great concern for all the food authorities. As all of you know, viruses are considered as intracellular parasites that are able to exploit the host machinery. Particularly important is a membranous system, which is clearly usurped by the viruses. And they are able to build real impressive structures from the membranous system of the cell. Here we have some examples that they are widely studied, and you can get them more information in the bibliography. But there are some impressive structures from the from the endosomal reticulum, and also from the mitochondria. One of the main actors in the membrane system reformation is the endosomal sorting complex required for transport, or ESCRT, and this complex is mediating some restructuration or some reshaping of topologically similar membrane-related processes in the cell. Here you can observe that some processes related with endocytosis or multivesicular endosome formation, specifically the formation of the intraluminal vesicles inside of the multivesicular body, is regulated by the ESCRT complex. But not only these kind of events, but also the ESCRT complex is regulating the cytokinetic abscission as well. As I commented before, the viruses are able to exploit this system because it's a great deal for them to be able to exploit the complex that is in charge of the membrane remodeling and reshaping. So, for instance, this complex is widely studied in the case of the HIV, which the virus is able to recruit some of the proteins of the ESCRT complex thanks to the interaction with the viral proteins. But also the herpes virus is able to recruit this complex to perform the exit of the nuclear envelope. Here you have some details of these kind of events, and also we are going deep in the structure of this complex, but basically it's kind of these filaments recruited in the place where the vesicle is gonna pinch off towards the cytoplasm. In this case of the HIV, with the GAG protein is interacting with some of the ESCRT complex proteins for viral binding. But here you have the example of the dengue virus, which is able also to support the replication and formation of the special structure to support the replication in the ER. But also the hepatitis C virus is also recruiting and exploiting this complex to perform this assembly sites. It's much less studied, but there are some increasing reference about the DNA virus and the interaction with the ESCRT complex. And here we have some of the viruses that are published that use the ESCRT complex for the infectious cycle supporting. Here you have one of the most known example, which is the herpes simplex virus, which is able to go through the nuclear envelope thanks to this complex and acquire one of the envelope, but then fuse with a membrane of the ER as well, and then get some of the tegument, some of the membrane of the virus to finally go out of the cell. And all of these steps are regulated for the ESCRT complex, which are basic for the viral cycle of this virus. But also one of the well-studied viruses that needs the ESCRT complex is the vaccinia virus, which is recruiting the ESCRT complex for the viral egress. not only viruses, but some other parasites that are using the ESCRT complex to build the replication complex. Here we have the example of the Toxoplasma gondii, but also bacteria survival and maintenance of the replicative compartment and release toward the membrane plasma. The plasma membrane is using this machinery, this specific machinery of the cell. Here you have a table that is showing some of the ESCRT complex that are recruited, some of the ESCRT components that are recruited for different viruses that are in this case envelope viruses, and here you have the reference, but there are a lot of good reviews about this topic. So here this is just a reference as well, but we are going deep in the ESCRT complex structure now. So the ESCRT complex, it seems like a multi-protein consists in multiple protein complexes that is sequentially recruited and required for targeting, and they are required for targeting a nucleation of the different cargos or ubiquitinated cargos, that are ESCRT 0, it's called ESCRT 0, ESCRT 1, and ESCRT 2. And this is gonna be used to recruit the ESCRT 3 component that it's formed this kind of filaments surrounding the point of the vesicle where the vesicle is gonna suffer the excision mediated by this ATPase, which is called BPS4, and this is an accessory protein. So to sum up the slide, which is a bit complex. So we have some of the components that the whole ESCRT machinery, and we have some different subcomplexes. One of them is ESCRT 0, the other one is ESCRT 1, and the other one is ESCRT 2, and all of them are going to serve to recruit the other components of the ESCRT complex machinery, which is ESCRT 3, the ATPase BPS4, which is going to be the responsible to perform the excision of the vesicle of the mediated for the ESCRT complex, and it's interesting to focus in some ESCRT accessory proteins like ALIX, because it's going to be quite exploited by viruses because it has the capacity to bypass the ESCRT 2. ALIX is able to interact with the ESCRT 1 and the ESCRT 3, and the part of the ESCRTs that are mandatory for this membrane processing is ESCRT 3 and BPS4. So ESCRT 1 is going to be accessory and ESCRT 2 is going to be accessory because the virus has the capacity to get the ESCRT machinery absorbing the protein ALIX, for instance. Here you have some schematic of how the ESCRT complex is recruited toward a point of the cell where a process is going to happen. So here we have some of the proteins that are able to be the target for the rest of the ESCRT machinery. So for instance, and this is depending of the process that we are paying attention. So this is ESCRT 0 is essential for the recruitment of the ESCRT machinery in the multivesicular bodies for the intraluminal vesicle targeting of the cargos. CYP55 is essential for the cytokinetic excision. GAC proteins of the HIV are essential for the budding of the virus. But here you have how these proteins are going to be necessary to trigger the assembly of the rest of the complex. Here you have the ESCRT 1, ESCRT 2, ALIX, and HTPTP proteins. And when we have this complex, the rest of the ESCRT complex are going to be recruited in form of 5 filaments, as you can observe here. This is the ESCRT 3, which is in charge of reshape the membrane with these kind of filaments that are triggered with the polymerization of the different subunits of the ESCRT 3 components. I'm not going to be in detail of this part, but here you have all of the proteins that are form the ESCRT 3 complex. So then finally, with this vesicle is invaginated, the BPS4 ATPase is going to be recruited by ESCRT 3 and it's going to resolve the vesicles toward the inside of the endosome in this case. So our question is, African swine fever virus infection cycle is dependent on ESCRT, because as we have observed in the first slide, is the ESCRT complex is the master machinery regulating the reshaping of the membranous system of the cells. So this virus relies in several and different parts of the virus cycle in membranes. Here we have how the virus is endocyted by clathrin-mediated endocytosis, but then it traffics through the endosome pathway, but then after a short phase in the nucleus, it binds the replication complex. In a perinuclear zone is going to construct the viral factory, and it's published by our group and others how this virus is able to recruit the membranous system of the cell toward the replication site. As we can observe here, this is the things that I mentioned before. Here we have some results published for me in this case, this is the aspect of how the vesicles in the cells positive for endosome, early endosome marker, multivesicular bodies, late endosomes, are lysosomes that are widely distributed through in the cytoplasm, how it changes actually after the virus start the replication and form the viral factory here in red. So here we have how the endosomal early endosome markers are recruited toward the viral factory, but also the CD63, which is a marker for the multivesicular bodies. Also the RAP7 positive vesicles are able to be recruited by virus toward the viral factory, but also the lysosomal marker are able to be here. So we think that maybe in this strong and deep process of reshaping of the endosomal or membrane system, the ESCRT complex could have a role. Another important and relevant stage in the viral cycle is the virus budding, which is performed by Philopodia, which has actin inside. And here the virus is in green, the actin is labeled in red, and here we can observe in this electron micrography also how maybe here, similarly to GAG protein of the HIV, the African swine fever virus could be able to recruit endosomal, the ESCRT complex machinery to this point to facilitate the scission. So these are our focus to perform this work. So I'm going to talk a little bit of the African swine fever virus particle. This virus is the only member of the ASFV family. It's the unique arbovirus, which is DNA, and it's a very big virus. It has 175 to 215 nanometers, and it has a nicosaedral symmetry, as you can observe. And as you can imagine, it's a double-stranded DNA virus with a huge genome. It ranges between 170 and 190 kilobases, and it has like 150 proteins. So this is a very complex virus. And to pay attention, because as I mentioned in the beginning of my presentation, this virus is a real concern for all the food authorities around the world, because the only thing that we can do to contain this disease when there's an outbreak is isolate the infected animals, but also the sacrificing the animals as well. So it has a really it's a real disaster for the economy of this industry. So we started. We thought that maybe it was interesting to cut the ESCRT pathway by silencing the last component to be recruited on this machinery, with which I mentioned before, the ATPase BPS4. So we pay attention to BPS4A, and we got a nice silencing of the protein. Here you can observe in this graphic as well also the levels of the BPS4. We observed that by analyzing the expression of the P30 protein, which is an early protein expressed at early stages in the in the viral cycle. In silenced cells, we don't get any effect, but we have a significant effect when we pay attention to a late viral protein, which is called P54, which is expressed during the replication of the virus, and we have here this significant decrease, but also in the amount of the DNA of the virus. So we have an effect, not an early stage, but we have it in the late stage of the virus. And also we analyze the viral titer, and we have some effect in the extracellular viral titer. So we have here, to sum up this slide, that we have some effect in the infection with we silence the BPS4A. As I mentioned before, there was an effect in the endomembranal system in infected cells, but this was not the case. The silencing of the BPS4 protein didn't trigger any effect in the recruitment of the membranes in infected cells. As we can observe here, we have this marker for the early endosomes, so we can observe that in infected cells, silence it for BPS4A, still we have this recruitment, and we have observed this similar pattern of the recruitment surrounding the viral factor in the case of the multivesicular bodies, and also in the lysosomal marker LAMP1. Also, we wanted to know what happened with other ESCRT proteins under the silencing condition of the BPS4, and we label this HRS component of the ESCRT1. We observe how the signal of this protein is slight weakly increased, but we observe here it's not an effect related with the virus infection, because here in mock infected cells we have this increase of the HRS expression when we silence the BPS4 protein. Also, we checked an important protein, as I mentioned before, ALIX, which is an accessory protein that many viruses brought to get any advantage for use the ESCRT machinery in its benefit. And here we have how the ALIX expression increased under the African swine fever virus infection and is not depending of the silencing of the BPS4A protein, because here in mock infected cells we don't get an increase that we in fact observe in the similar condition but in infected cells. And also another important point here is that ALIX was recruited toward the factory, the viral factory, in both conditions, in control conditions and in silencing of ALIX protein conditions. So we have this protein surrounded and in contact with the factory, and we observe here in this graph of the quantification of these images. Next, we try, in the light of these results, we thought then we are going to silence ALIX expression to check what happened with infection. So here we can observe how we got this nice silencing of ALIX protein, and in this case, again, we have that we don't have effect in the early stage of the viral cycle, but we have it in the later stages of the viral cycle. We checked for this the expression of the late protein P54, and also we measure the amount of the viral DNA, and we have this decrease. Also, we have the effect in the viral titer. So we have an effect of the silencing of ALIX protein in the viral infection. And the other thing that we would like to check is that, importantly, ALIX is able to interact with a rare lipid, an unconventional lipid, which is called LBPA. LBPA is a lipid which is located mainly in the late endosome, and ALIX is able to interact with LBPA in a calcium-dependent manner. And LBPA and ALIX do that to be able to form also intraluminal vesicles in the multivesicular body as well. So it's a kind of different mechanism for formation of intraluminal vesicles than the canonical one that is regulated for ESCRT, and it was kind of surprising for us when we saw that ALIX has a role, and then it directs us to check that if LBPA is able to perform any role in the infection of the virus. But interestingly, LBPA is not only able to interact with ALIX, but also LBPA is able to interact with many other relevant, to participate in many viral relevant endocytic events. This slide pretends to resume these pathways. Maybe it's a bit messy, but I'm gonna be focusing the important points here. Here we have the LBPA, which here is called BMP because it has this other name also. As I mentioned before, LBPA is located in the late endosome. So here we have the late endosome. Here we have another late endosome, and as I mentioned before, LBPA and ALIX are gonna interact to form these intraluminal vesicles that are important, according to the bibliography, for other viral infection like VSV, but only some for the infection of this of other viruses, as I mentioned, VSV, because this virus VSV is gonna go out to the cytoplasm by a process which is called back fusion, and this back fusion is really specific, and it can be triggered by the LBPA and ALIX in the fusion with the limiting membrane of the late endosome, and this fusion is regulated by LBPA and ALIX. So maybe this is an important point of interest of our virus, not only for if the African swine fever virus is suffering back fusion with the late endosome, that we don't think so, because this virus is huge to be able to go in the intraluminal vesicle, but maybe it's important in other step in the infection. And also here we have another important process for African swine fever virus, because LBPA in green, repeat in green, is able to interact with the proteins that are able to efflux the cholesterol of the cellular cholesterol, because LBPA is able to interact with MPC2, which are gonna give the free cholesterol to the protein that is able to efflux the cholesterol out of the cell to other regions of the cell, and this is called MPC1. So in this machinery in charge of efflux the cholesterol out of the cell to our cellular destinations, MPC1 and MPC2, LBPA is able to regulate this process by interacting with the protein MPC2. And we published a paper that this efflux is essential for the virus infection. So we have an actor, which is LBPA, that is able to regulate the intraluminal vesicle formation in the multivesicular body, but also the back fusion in the compartment, and also the important process which is called cholesterol efflux. LBPA is also involved in this step. Importantly, MPC1 is the intracellular receptor for Ebola virus, which has a very, very similar viral cycle that African swine fever virus. So in the light of this slide, we maybe you are able to understand a bit better maybe the possible role of LBPA in this virus infection. So first, what we do is, by using an antibody that is widely referenced to block the LBPA function, we perform we incubate the cells with the antibody, with this antibody anti-LBPA, and then we check that if the antibody was correctly injected into the cells, so we can be sure. Here in green, we can be sure that the function of the LBPA is impaired, and surprisingly, we saw how the early viral cycle step was affected. So maybe in the entry, or maybe in the step of that was related with the endosomal traffic of the virus through the endosomes, but also the late stages in the viral cycle are also affected, like the P54 protein expression, which is expressed in the replication of the virus, but also the DNA amount of the virus. Here we observe that we perform an immunofluorescence, how in this image the LBPA is also recruited toward the viral factory, which is here labeled in red, and you can see the whole panel. In here, this was measured, and we can observe how the recruitment, in fact, the recruitment of the LBPA intensity toward the perinuclear region, which is the place where the African swine fever virus builds the replication complex. So, as a conclusion of this study, we can affirm that BPS4 silencing impacts late at the viral cycles, affecting African swine fever virus replication, viral progeny formation, or budding. This would suggest a potential role for this ESCRT protein in African swine fever budding. African swine fever virus weakly increases HRS expression. Thus, this requirement of HRS and African swine fever virus infection is not yet well understood, since it traffics through the endosomes independently of intraluminal vesicles. African swine fever virus infection overexpressed and redistribute ALIX to the viral factory independently of BPS4A silencing, supporting later stages during the viral cycle as replication, viral production, or viral egress. LBPA blocking regulates African swine fever virus infection at early and late stages of the viral cycle, probably by affecting viral entry and fusion, by impairing cholesterol efflux from late endosomes. LBPA is also redistributed to African swine fever virus assembly sites, supporting a function in viral infection at later stages. At the moment, these are our conclusions. We are still working in deciphering the possible involvement of this machinery in the African swine fever virus infection. And finally, I would like to say thank you to all of you. Really sorry that I'm not able to share this time with you there. And I want to present my team. This is my boss, Covadonga Alonso. Also, we have the Isabel García Dorival, which is also a junior investigator. This is me, Miguel Ángel Cuesta, Lucía Barrado. We have a PhD student, who is Paula Gil Cortés, and a postdoc, Inmaculada Galindo, Ana del Puerto del Pino, and also another PhD student, who is Jesus Urquiza López. I want also to say thank you to all our collaborators of the VIX unit, African Swine Fever Virus Interactome Consortium. We are funding also for La Caixa, and yes, also we have our really good collaborators as Carmen Hill, Fernando Diaz, and Oscar Fernandez at the CIV, and also César Muñoz Fontela at the Bernard Nock Institute, and Professor Miles Carroll, and Professor Stuart Armstrong and Julian Hiscott at University of Liverpool. So yes, thank you very much for your attention, and I hope you enjoyed my presentation.